In this chapter, we will deal with gradually the data types, the data structure that will be present in the rest of the course. Very important always is to clearly understand the terminology and sometimes clearly understand the confusion there is in terminology, because different people may be using the same term for a different reason, for a different concept, or they may use two different terms but apparently are talking about the same thing. One thing is clear: everything we do in this course implies outcomes, y variables, in other words, dependent variables that are in one way or another correlated. Now that can happen in many different ways, and the task of this chapter is to kind of overview these different ways, give a review of it, and then of all the key areas, give an example to make it a bit more lively. So if we start simple, the simplest problem we can imagine is the one sample problem. We have a collection of outcomes for a certain sample size, n, and those outcomes all follow the same distribution. For example, we have a group of people and we measure their body mass index. Okay, on that sample we can calculate a number of things, like we can calculate means, medians, and that's a discussion in its own right, of course, what to use in what circumstances. And you, as an applied statistician, as a practicing statistician, better has some working knowledge, some working models as to when is which technique a little better than the other. A frustration that especially more mathematically oriented people sometimes have is it's not crystal clear. There may be two perfect experts who have on a certain choice a different opinion. That's perfectly possible. There are also cases where it doesn't matter so much. If you have a nice symmetric distribution without the risk of outliers too much, whether you calculate the mean or the median may not be so different, of course. But there may also be considerations in terms of precision, the median being a little bit less precise than the mean. Okay. Next, when we calculate such quantities on a sample, of course, we should not forget that we're trying to estimate something for a population behind, the estimand, what we're trying to estimate. And therefore, we should realize always that the sample is a tool to get to that quantity with a certain variability, with a certain imprecision, a certain error. And therefore, we usually quantify the variability through standard deviations at population level, standard errors around an estimate for a population mean, for example, but sometimes also interquartile ranges or other non-parametric methodology. Now, if I talk about body mass index for a population, quickly the population will split into subpopulations, males and females. So we may have two groups, the two-group problem. G is equal to 1 for males, 2 for females, for example. And we have a sample of 100 body mass indices for males and females, and we can estimate the quantities from the previous page now twice, once for males, one for females. An additional effect may be, or an additional consequence of doing that may be, something that comes naturally, as it were, is estimate the difference between males and females. Do hypothesis tests as to whether males and females have the body mass index, formulate a hypothesis test. But at any rate, we are now talking about two variables. And I don't mean body mass index into two groups. No, I mean body mass index is Y, and gender or sex in this case is X. There is a covariate. Yeah, there are two groups, yes, but we can think of the groups as indicative of the two levels of my variable sex in this case. The two-sample problem. And of course, the two-sample problem is the basis, the core of many areas in statistics. Clinical trials is usually, not always, but usually centered around the two or more group problem. In epidemiology, I may be interested in the difference between two interventions. I may do an agricultural experiment with 2 fertilizers or 3 or 4 fertilizers that I compare. So it means this 2-sample problem is heavily researched. The 1-sample problem not, yes, also in a survey we often want to estimate a population quantity, but the design gets so complicated that we need to do a number of corrections for stratification, etc. But that would start to take us further. But the 2-sample problem really generates a large amount of designs and areas within statistics. What would we do with such data? Analysis of variance, for example, but the Wilcoxon test, a t-test, a variety of things can be done. If the outcome is binary, some people may start thinking of chi-squares, Fisher exact, etc. And then, of course, evidently, if you have a covariate, like sex, you can say, let me generalize that, and maybe in a different study, it's not going to be sex, but it's age, or time since the beginning of the study, or time since you took medication, or whatever. Or you may have other covariates that allows you to put the outcome, y, and the covariate x into a diagram, a 2-dimensional or a higher dimensional diagram. There's always one axis for the y, and there can be one or more axes for the various x's. And we get regression lines or regression curves, regression planes, etc. The whole regression area opens up for us at this time. Okay, again, you can do ordinary linear regression. There can be robust versions. There can be semi-parametric, non-parametric versions, for example, kernel density estimation. All sorts of things can be done here as well. Of course, I said a moment ago, there can be one covariate, sex, and that's a 2-sample problem still, or age, and then age, of course, is a continuous covariate, especially if you measure age very precisely. There will be as many different ages as there are people in your study. So that's a different ball game. But then I already hinted at the end, there can be more than one predictor. There can be sex and age at the same time. Now that all of a sudden opens, and I know we know all of that, but that opens the door to a variety of possibilities, but also a variety of issues. Because age and gender as covariates, the next question may be, yeah, but what about the interaction between age and gender? There can be an interaction indeed. So if you have 2 covariates, maybe or maybe not with interactions, or 3 or more covariates, we are talking about multiple regression, for example. Multi-way ANOVA, if all of those covariates are categorical or binary, then you may have, for example, treated, untreated patients, male and female, and that would give 2 by 2 is 4 different groups. It would be a multi-way ANOVA. But if all of them are continuous, or some of them are continuous, I mean the X variables, the independent variables, we talk about multiple regression. So we should, of course, recognize all of this in practice before we even select a technique to analyze our data. Otherwise, we may select a nice technique, but for the wrong problem, or we would be approaching our problem in the wrong way. Linear regression, yeah, but that's when the y is continuous. But no worries, if the y is binary, it would become logistic regression. If the y is a count, like the number of epileptic seizures, you could think of Poisson regression. And if the y is time until the patient cures, we have survival regression or time to event or reliability regression or whatever you want to call it. Again, once you have 2 or more predictors, life may actually become relatively complex, and there are entire areas of statistics or related to statistics like epidemiology that are looking at this. So just imagine I'm interested in an outcome Y, which happens to be lung cancer. Okay. And then I'm interested maybe not in a clinical trial context, but for example, whether in a certain industrial environment, the exposure to the chemical element cadmium is happening, yes or no, or to a certain extent. So my covariate, X, would be cadmium. But of course, I cannot do an experiment. I cannot say 50 people are exposed to cadmium, 50 people are not randomly allocated. No, that's unethical. I can do that in a clinical trial with a medication. I cannot do that with a harmful effect. So what is the solution? I use the data that are available anyway. There may be historic contamination, and I may have data available. But it may then well be that the people who are exposed and not exposed are different. They may belong to different socioeconomic strata. They may live in different areas and therefore are also exposed or not exposed to a number of other things. And in particular, I can think of smoking status. People exposed and not exposed could have a different smoking status on average. Well, maybe not. True, but maybe yes, and I cannot exclude that. So I should at least take that into account. So one part of methodology would say, well, this is just another X variable. X1 is cadmium, X2 is smoking, maybe there is interaction. But in an epidemiological setting, we say it's true, but cadmium, that's the exposure of interest. Smoking is a background variable that may well confound the relationship, because if smoking is related to cadmium and smoking is related to lung cancer, and we know that that's the case, and if we then don't take smoking into account, we may get the wrong result. We may estimate a result, a relationship that is not true, but is only seemingly true because we ignore a confounder. So what the pure statistician would call Y, X1 and X2, the epidemiologist gives very precise meaning. The disease, the exposure, and the relationship between the two, but which may be confounded by other variables such as smoking and several others. Now, before in this area data are collected and people agree on the model, it may take quite a bit of time. And the model at the end may be a logistic regression, lung cancer, yes or no. Severity of lung cancer may be a linear regression, etc. So we may be doing a regression at the end, but precisely how we use and code our covariates and which covariates that are, that becomes an issue of debate, because there are many variables at the same time. Now, many variables at the same time, that seems to indicate the theme of the course, multi, like multivariate, but not yet. We're not there yet, because I think it should be clear from this slide that even if there is a single Y, the design of the study may make it very difficult to, and open to debate and controversy, to decide what kind of model we are going to use. But still, we have a single outcome. And that may change when we have, of course, several outcomes, several dependent variables. We will call that in statistics multivariate methodology, but we have to be very careful. In the medical literature, if you would have, for example, a disease, let's say lung cancer, and then you look at the effect of treatment, but also age, gender, previous therapies, etc., and we account for all these X's at the same time, well, we statisticians, we call that multiple regression. But in the epidemiological and medical literature, you may often see multivariate regression. So there is a confusion. In the medical literature, multivariate means something else than it does for us. For us, because we need to distinguish between more than one X multiple, more than one Y multivariate, we do need the two words. This doesn't mean from now on we should fight against that use in the medical literature. No, the burden is upon us. We should be aware of that difference. And if we are in doubt, if you're in doubt, when I talk about something, when somebody else talks about something, when the medical doctor or your client talks about something, don't start guessing what they might perhaps mean. Ask. It's not a stupid question. It never is, because there simply may be different meanings attributed to the same term. So it's not a matter of being so clever as knowing what a doctor means. There's no way of knowing. So you should ask. That's the way forward. Okay. So in multivariate analysis, we are going to analyze more than one outcome at the same time. And to fix ideas, take blood pressure. If we measure blood pressure, you may say, I measure my blood pressure once through, but you will get systolic and diastolic blood pressure. Two values, higher and lower. So you naturally get two outcomes at the same time, which we then call a bivariate outcome. And naturally, these two are going to be correlated, right? You can be sure of that. Likewise, if you measure with children growing the length of their left arm and their right arm, very likely they will be correlated. Otherwise, you may, if that's not the case, some people may have a long left and a short right arm, or vice versa, etc. No. Nature, especially living organisms like even plants, many animals, human beings, are symmetric in one way or another. There are many axes of symmetry, which means that the measurements are extremely highly correlated. Now, correlated, I can say they're dependent. I can say they are associated. And this is also very important, especially in a course like this. We should be very clear what we mean by the term. Association and dependence are more or less synonyms. But dependence means that's the absence of independence, the reverse of independence. So there's the mathematical concept of dependent and independent random variables. Association kind of focuses a little more, and it's subtle, focuses a little more on the fact that that dependence can be quantified through measures described by measures of association. Like, for example, correlation. Like, for example, odds ratio. Like, for example, Kendall's tau. So all of those are measures of association. So association and correlation are not synonyms. Correlation is a precise measure of association. Association is more a synonym of dependence, but naturally leading to the concept of actually quantifying that dependence, that association, in one or other way. And then when we talk about correlation, we should be very clear and careful what we mean. Do we mean Pearson correlation between two different but dependent random variables like systolic and diastolic blood pressure? Do we mean the more non-parametric version of it, Spearman's rank correlation, the rank-based version of it? Do we mean intra-class or intra-unit correlation for cluster data, where I have several outcomes that are clearly correlated, but where I cannot say this is the first one, that's the second one, like, for example, in a group of fetuses that all belong to the same litter in a rat or a mouse experiment. Every two litter mates may be correlated with a common correlation, but without being able to say, well, these are all the first fetuses and these are all the second fetuses. No, it's like a bunch and there is no real structure. It's a bit amorphic. So that's what we call intra-class correlation. Coming with many other names, intra-familial correlation, intra-household correlation, because many people will call that intra-something correlation, and the something will be relevant for their problem, and that may be different from your problem, for example. Okay, good. That brings me to the first example, the multivariate example, and not only because it will be used in the course, also because it's one of the earliest multivariate data set, and earliest, I now go back about 100 years ago at the time of Fisher and Pearson. It's sometimes called the Fisher iris data set. So, What is it? We have iris flowers, and those iris flowers are measured, are recorded through 4 variables. The petal or petal lengths and widths. So, and then the sepal lengths or width. So there are 2 types of leaves. The leaves on the flower and the leaves on the stem of the flowers. So the colored ones and the green ones, so to speak, and they have a length and a width. And you can imagine, like any body characteristics, that also 4 characteristics on the same flower will somehow be dependent. Okay. In this case, there are 3 different species. Each species is replicated 50 times in the data set, leading to 50 setosa, 50 versicolor, and 50 virginica. So, clearly, the length and the width variables is the Y group. So there is a Y1, Y2, Y3, Y4. It's 4 variate, multivariate, but if you want to be specific for this example, 4 variate. And then the question is, how many covariates are there? Well, some people say 1 or 2 or 3, and it's all right. 4, no. But you may say, well, I see a list with 4 Ys, Y1, Y2, Y3, Y4, correct. And then I see a list with 3 stars, so it must be 3. Not necessarily, because there is actually 1 nominal variable. The X variable has 3 levels: setosa, versicolor, virginica. Some people will say, yeah, and because it's nominal, I want to replace it by 3 dummies. So, X1, X2, X3. And then somebody smart comes and says, yeah, but if you have 3 dummies, 1 is redundant. Because if you know that a flower is not setosa and not versicolor, so it's 0 on both of those, you know it must be 1 on virginica. So it's redundant, true. So there are 2 there are 2 linearly independent indicator variables, 0, 1 variables. There are 3, but then 1 is redundant, or you can say, I rather consider it a nominal variable with 3 levels. Be my guest, that's fine as well. Okay, so this is a brief snapshot on the data. It's not 150 lines, but just a few every time. So you see the species indicator in the same ordering as they were listed here, setosa, versicolor, virginica. And then we have sepal length width, petal length width. It's very important, if you present a data set, to be aware and, if you can, report the units. So this data set is now reported in millimeters. There are versions of the IRIS data set where the reporting is in centimeters, which is of course exactly the same, except 33 will become 3.3. While this is not specific to this course, evidently, if you reach conclusions, put units next to them and check for yourself, before your audience does, whether the numbers make sense. So if you say the average length of the sepal leaves in the IRIS data set is 743 millimeters. Well, you would have discovered a gigantic species of iris data sets, of iris flowers. They don't exist in that form. If you say, on average, people in this study get 752 years old, that may be biblical, but human beings don't reach that age. So units should be reported, and the sense or nonsense of your conclusions, and your data for that matter, should be examined. This is a brief graphical representation of the IRIS data set. So along the diagonal we have box plots showing a certain amount of symmetry and giving you a sense of the variability. And the off-diagonal plots are actually pairwise scatter plots. Remember, there are 4 outcome variables, and therefore it's not a surprise to see 1, 2, 3, 4, 5, 6 of those pairwise plots. Clearly, we see that in some cases we have separated islands. It's like a sea in which you have two countries, two islands located. And that's, of course, not a surprise because there are three species. You may well see that the data split into three or more. Even if a species would split further, in that case, you would see more. But we actually seem to see two. And a preview of some things that are going to come later is, yeah, there are two, and that's because two of the species are actually touching and slightly overlapping. Not a lot, but they do overlap. Well, it's also clear that you clearly see that in some graphs here, and of course this one is the same but symmetric, and also here, but somewhat less clear in others. So the separation between species is not seen necessarily in the same way in all subpopulations, which is why if you have the question to separate your populations, to somehow see the difference as clearly as possible, that you may have think of ways of doing that best. Do you need all four variables, or do you choose one or two that separate best, or do you do something else? So if I want to clearly see, describe, use the difference between the species, how am I going to do that? That may be the scope for discriminant analysis, and discriminant analysis will be mentioned later. And later is now, because what questions can I ask indeed? Are there differences between species? Yeah, we were looking at that, we do see them. But of course, how do I best see them? And are they significant? I may want to do tests for that. Given a species, do we need all four variables to have an idea about the characteristics? So how can I characterize a species? It's not because I measure 75 variables, it's only 4 here, but let me exaggerate. It's not because I measure 75 variables that I need all of them to see the difference best. And then, what is now the best way to capture the relationship between the sepal variables and the petal variables? Or likewise, is there a relationship between the lengths and the widths of the leaves? Now, if I would reduce that to a single variable, is there a relationship between sepal length and sepal width? You calculate the correlation, for example, whatever correlation you have in mind, fine. The relationship between a single variable on the one hand and several variables on the other, so a y and then an x1 through an xp, that we have done too. And in case you doubt, multiple correlation. The coefficient of determination, the R square in regression, is exactly that. The relationship between an outcome variable and several predictors. But now we go one step further and we say we are interested in the relationship between y1 through yq and then x1 through xp. In the iris data set, p and q are equal to 2, but that number can be arbitrary. So what is the relationship, for example, between systolic and diastolic blood pressure on the one hand, and age, gender, dietary habits, sports habits, drinking habits, and whatnot of the patient? 2 versus 10 variables, for example. That will be the playground of canonical correlation. Okay, so as I said, for a univariate sample, a difference can be estimated quite easily with a test for the 2 groups or the 3 groups. But then, if I have sepal length and I have 3 groups, do an ANOVA. A 1-factor ANOVA with 3 levels for that factor. Fine, you're done. But if you have sepal length, sepal width, petal length, petal width, is there a difference on each one of them? On all of them? More on one than onto the other? And where do I see it best? So the floodgates of all situations are open now, and we want to look at that in an orderly fashion. So, do we need all information to look at the data? Let's put it very informal. Well, that's the playground of principal component analysis. If I have a single variable, normally distributed, it's characterized by its mean and its variance. If I have 10 variables, there are 10 means, 10 variances, and there are 10 choose to, which is 45 correlations. So you see with dependent data, not only is mean and variance supplemented by correlation, also the number of correlations increases quadratically with the sample size. Okay, so that's when I just want to look at my population and maybe I want to reduce the amount of information as much as possible without losing too much information. Reduction is throw away as much as possible. Without losing information is keep it all. And these two cannot happen together, so I need to find a compromise between the two. Next is, can I reduce it in such a way that I still see the differences between groups optimally, and I can actually create variables that really maximize the spread, etc. That will be, on the one hand, the playground of discriminant analysis. How can I maximize the view on the difference? And then factor analysis is kind of in between, because it's also like principal components, but taking covariates into account. So in discriminant analysis is, okay, I have a new iris flower. There is the data set with 150, but now there is an additional one, number 151. I'm telling you petal length, petal width, sepal length, sepal width. I measured those 4 characteristics. But now the question is, which one is it now? Is it setosa? Is it virginica? Is it versicolor? And, well, the goal of discriminant analysis is to come up with an optimal rule, a function in which you put your 4 measures and you get out the best possible classification. Well, what people like us statisticians to do is to tell them, this is certainly setosa. We may not be able to do so. Not because we don't know our job too much. That's not the case. No, basically because a perfect classification is not possible. So in discriminant analysis, we don't look for the perfect rule, but we look for the best rule. The best rule may not be good, but that's because the data we have to work with, the raw material, is not in a position to discriminate a lot. If I tell you, I have a human being, adult, Caucasian, for example, and the person measures 1 meter 68 centimeters, male or female. Well, that should be pretty hard, because in that range, there are relatively tall females and relatively short males that overlap in that category. But if I tell you somebody is 2 meters and 5 centimeters, you may tell us, I'm pretty sure that that's a male, because it's exceptional but possible for males. It's very, 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 very rare for females, etc. So you get the point. The variables used for discrimination may or may not do the job very well, and depending on the numerical value, I may be more in a position to make error than others. And that's what medical doctors, for example, are confronted with all the time. They measure a number of things on the patient, and then they should decide, prescribe medication, yes or no, refer to a specialist, yes or no, refer to the hospital, yes or no, call in the ambulance because it's so bad, yes or no, etc. All of these things are part of the discussion. And then last but not least, I already said, if you have a group of variables on the one hand, like the petal leaf variables, and a group on the other hand, like the sepal leaf variables, what is the relationship between the two? That's canonical correlation. Canonical correlation will not be treated in the course. Principal components analysis and discriminant analysis will. So, the general multivariate setting is, we have a set of dependent variables. You have to have 2 or more dependent variables. Otherwise, it's not multivariate. What about the independent variables? Anything goes. You can have 1, 2, or more. Maybe sometimes there are no independent variables. There is just the 4 length and width variables, for example. That's all we have. If let's say we have 200 replicates from Setosa, not from the other species, it's a homogeneous population, but yet we may want to study the interrelationships. But the bottom line is we need more than 2. What outcome type? In general, everything is possible, but studying the very general setting all at once, that is, of course, going to be very difficult. What are other correlated data settings? Well, you can have, for example, systolic and diastolic blood pressure. That's bivariate. Systolic, diastolic, plus high density and low density lipoprotein, that's clearly multivariate. But if we measure all those things more than once, it becomes repeated measures-ish. Let's focus on blood pressure. So let's say I have systolic blood pressure and diastolic blood pressure on patient I. But now I'm measuring that 3 times. I'll indicate with J the replicate. So I'm measuring your blood pressure, systolic and diastolic blood pressure, at 2 o'clock, at 2 minutes past 2, and at 4 minutes past 2, because I, as a medical doctor, the general practitioner, I want to put you at ease. And I want to make sure that by the 3rd measurement, you're so bored that you're totally calm. That's the idea. Well, if I then see a downward trend, just because you're calming down a little bit, you're a little nervous, you're going to the doctor and you couldn't find a parking place and you didn't know the address well and you hadn't seen that doctor before, etc., etc. But after 3 measurements, you're calm, they seem to go down. I'm not going to draw any conclusions other than that the 3rd measurement is under more quiet conditions. Of course, if I measure your blood pressure, every 6 months for 10 years in a row, those 20 measurements will be your individual blood pressure profile. And now I can start to draw conclusions. Do you evolve upward, which is not a good thing, but that's human nature, that's what happens, that's biology, but more rapidly than the average of the population? That's bad news. If you don't increase over time, you're always staying the same. Good news. You're increasing, but not as fast as the population average. Better news than on average, for example. So then I may start to draw conclusions about the evolution for you in particular. And if I look at all the data I collect together, I may start to draw conclusions about the population we are studying and we drew the sample from. So measuring the same thing or the same things more than once, we call that repeated measures. But when the time ordering becomes relevant in its own right, because I can give you, I can study the decade-long evolution for somebody between 40 and 50 years of age, that we call longitudinal. And then if we take it one step further and we say, yeah, but there are other quantities that we are measuring not in time, because longitudinal data means the measurements are taken in time. So whatever I'm looking at, let's say systolic blood pressure is measured at certain time points, and I create an observed profile from which I deduce an ideal profile. That's what the modeling will do, a clean ideal profile behind it, longitudinal data. But the point is, diagrams like this are very intuitive and simple and natural, because time is unidimensional and it's directional. It flows from the past to the future, not the other way around. Of course, when I talk about time as a dimension, you will tell me space is another set of dimensions. For example, I may be interested in the abundance of ore, like gold or silver or diamond or whatever, and therefore I start exploring, drilling or whatever, oil, in various places. And I don't only know down the abundance, I also know down the longitude and the latitude, the x and the y coordinate, until somebody comes around and says, yeah, I know the coordinates now, but how deep is it? Because if it's, and I'll exaggerate a little bit, if it's 10 meters deep, or it's 10 kilometers deep, that's a different story. In the second case, it may be there, it may be very abundant, but commercially completely uninteresting, because there is no point in clear and profitable exploitation of that resource. So depending on the problem I'm focused with, I may be interested in the data connected to their X and Y location on the surface of the Earth. I may also be interested on the depth or the height in addition, and then I have, rather than a two-dimensional spatial data set, I have a three-dimensional spatial data set. An archetypical example of a two-dimensional spatial data set is for every county or province or department or whatever, I have the number of children that are born with birth defects relative to the total number of births, of course. So the information I may have will be a particular point in a county, the county seat, the county average, whatever, the point of equilibrium, and the variable of interest and maybe some covariates. Okay. Now, that means that two-dimensional data is relevant. Three-dimensional data in a population setting like this may not be relevant except in very mountainous areas. In Switzerland, for example, I may not only record the X and the Y of the village, but also the altitude of the village, because a village and a nearby village may be at almost the same location in X and Y terms, but there may be, in terms of altitude, 1,000 meters in between. And it could be more in certain mountainous regions. Is univariate data or unidimensional data spatially possible? Yes, of course. In so-called line transect data. In line transect data, we are interested, for example, in animal abundance. There are reports that the number of insects the insect population is drastically reducing. Is it true? So we take a large natural area, and a green area, and we randomly throw a line on that, with a computer, of course, on that area, and along that line we go and we count everything that comes in our line of sight. That is line transect data. And then cluster data is when data are correlated or associated or dependent, but without a clear ordering structure like time and space or time and space together. For example, fetuses in a litter. When a rat is pregnant or a mouse is pregnant, it may produce between 2 and 16 litter mates. Okay, these are correlated, but no clear structure can be discerned. It's not like we have the first and the second and the third fetus, etc. May be dependent on birth order, also the position in the uterus line, so sometimes it is relevant, sometimes it isn't. Okay. So this is a variety of settings where dependent data occur, and again, there are no clear, hard distinctions between these areas. They sometimes happen together, in other words, like systolic and diastolic blood pressure is bivariate, but if I measure them repeatedly over time, I actually have bivariate longitudinal data. If I collect data on river pollution, I may be interested in space and time. Where on the river am I measuring and at what time? So, for example, what is happening here may be happening downstream a while from now. Hurricane paths is an example of spatio-temporal data, etc., where space and time are of interest at the same time. And then, whereas in general space is not ordered, rivers do give a certain unidirectional ordering to space, because rivers flow in one direction at any point in time. Many rivers do that all the time in the same direction. Land rivers, as they're sometimes called. Sea rivers or tidal rivers is that part of the river that is subject to the tidal activity of the sea. For example, the river Scheldt in Belgium, up from the coast up to Ghent, is tidal, meaning that the water flows half of the day upstream and half of the day it flows properly downstream, because rising sea levels make the water flow inland, and then later it flows out again towards the sea. Okay. So here are a few examples, and we should now do this little exercise to recognize which they are. Look at this table and make up for yourself which situation we are covering here. For each subject in a study, height is recorded. Well, that is clearly univariate, of course. It's classical univariate. For each family in a study, the height of all siblings is recorded. Well, that's cluster data. And for each subject in a study, height and weight is recorded. Well, that's clearly multivariate. Okay. So, it is clear that variables that are correlated may be more or less homogeneous. For example, high-density lipoprotein and low-density lipoprotein, they may well be measured in the same units. Systolic and diastolic blood pressure may be measured in the same units, right? It makes little sense to measure one in millimeters and the other in centimeters. And even if we do, it's not a terrible problem, but it would be a bit strange. However, if you measure the two at the same time, you may not have the same units. And you may add, for example, CD4 count, which is the number of cells per cube millimeter blood, or something like that. So it actually means that some variables that we study together intrinsically cannot be put on the same scale, because some are in centimeters, some are in liters, some are in kilograms, some are in compound units. Which also means that graphically displaying their profile makes very little sense. So if I say for a subject, this is systolic blood pressure, that's diastolic blood pressure, H density, low density lipoprotein, and I create a graph like this, this is utter nonsense. It makes no sense whatsoever. I can only do that when things are comparable and in the same units, like growth. If I take children growing, I may get a growth profile like this, with a genuine meaning, every time I'm measuring the same thing, and it always means the same thing. Okay, so for example, if I have 3 growth measurements, you would think 3 measurements, that's 3 means, 3 variances, and 3 correlations. Well, maybe you say, yeah, there are 3 parameters a priori, but if I graph it for this particular organism over this particular time frame, it looks like a straight line. And a straight line is simpler than 3 parameters. You can describe it with 2. And maybe the correlation between all measurements, the same number of units apart, or perhaps even all of them, the correlation is constant. There are 3 correlation parameters which I can simplify into a single one. So what is the message? If you have highly structured designs, like longitudinal data, repeated measures, spatial data, because of the structure, I can sometimes simplify my models. Now, not always. Some growth curves, as I was just drawing, would form a fairly highly complex nonlinear profile. And then the question is, shouldn't you leave it unstructured? And if you don't leave it unstructured, shouldn't you think of a complex nonlinear model outside of the scope of this course, of course? But in other words, the bottom line is the structure can be used. Okay, good. So what will we focus on in the remainder of the course after all the examples have been introduced? Multivariate methods through the techniques that I mentioned, principal components, discriminant analysis, longitudinal data and cluster data, making a distinction between them, but yet putting them in the same framework. And then a brief digression into spatial data. Longitudinal data and cluster data will be treated both for continuous as well as for non-continuous outcome variables. And spatial data will be treated in more detail in a different course on spatial data, so it will only be touched upon at the very end of this particular course. The longitudinal data and cluster data also return in the course on longitudinal data analysis, where more models and an in-depth treatment of the models considered here will also be given. Okay. That is the first part of chapter 2. We will now terminate this session, and then in the next web lecture we will continue starting from section 2.7 on the longitudinal and other examples.